right, it is 12:30, everybody, so we will get started. So before we start talking about Chapter 6, just a couple of reminders for everyone is your paraphrasing practice is due tomorrow. I just want to show everyone where it is on Brightspace in case there's any confusion about it. So I have the Brightspace page pulled up. It should look like this for you and me as well. So if you go to the Welcome to Biochem module here, there is a section of it called Research Project, and in there is a discussion forum called Paraphrase an Article. So your next few assignments will all be in here. So the one tomorrow is your paraphrasing practice. So here you're going to add a new topic to the discussion. You're going to say an article that you chose that relates to your particular research project. You're going to link us to your article, and you're going to include an original quote from the article as well as your paraphrase of that quote. About a week after that, everyone will have to respond to everyone else's paraphrase. So you'll pick someone else's post in the forum. You'll say either that looks great, or you can say, hey, like maybe you should define what this abbreviation is, or maybe, this was too close to the original. You should maybe change the wording a little bit. Or if someone did really do a great job, instead of just saying great job, you can all take your turn at paraphrasing their paraphrase. So get from a different angle or just to sort of reiterate what they were saying. So those are, yeah. Oh, okay. Yeah, and then the third one, not everyone will have to do, that's the paraphrasing revision. So after your paraphrasing response, you're going to get a score eventually from me about a week after that's due. And if you have a meets or exceeds score, you're good. If you have a no approaches or fails to meet score, you will have to do a revision of your original paraphrase. Just try again. And so not everyone will have to do that revision. So that's going to be where your next few assignments are relating to your research project. And I just want to, it says this on here too, but if you're working with a partner, you still have to do this part individually. You can't do this with your partner. I want posts from every person in the class. Any questions about the project? That's paraphrasing stuff. Yeah.
So it's a paragraph or paraphrase that whole paragraph? One line.
Yeah, I would try to do more than a sentence. Do like a paragraph or a few paragraphs. It doesn't have to be like the whole article. A lot of the research articles you'll find are very long. But yeah, if you want to pick anywhere from a paragraph to maybe like a page or so of it, just to paraphrase what it's about. Then this is on the Brightspace page. So if you go into the Welcome to Biochem module and scroll down, there's a submodule called Research Project. You might see it collapsed like this. If it's collapsed, just click on the little arrow to open it, and then you'll see this discussion forum, the paraphrase an article right here. Yeah. Yeah, so it's like a discussion forum. So you can say like add new post, that kind of a thing for your original paraphrase. And then when you respond to someone else's, you can do it as a reply to theirs.
Okay.
And there should also be a link to this in this week's. If you go to this week, you don't want to look. I put a post on the top right here to the paraphrasing practice. So any other questions about this? All right, so let's talk about chapter 6. Oh, okay, yeah. That's a good question. I guess it sort of depends on the original. You want to get across what they're trying to say, but in your own words. So it may be as long as the original. It may be shorter. It probably won't be longer. But it depends, I guess, on how dense that information is. Like maybe there's something in there that you feel like you want to unpack more, or there's something in there that you feel could be summarized down. It depends on what you're paraphrasing. I don't have like a length I'm looking for, if that answers your question. Yeah. Yeah. Global structures. Local structures. From
the first slide.
Maybe. So Where are you seeing local? Right here. Ah, okay. So yeah, local means it's close to each other in one area.
So it's just very simple.
Yeah.
It's there, it's local.
Yeah, like if your protein amino acids that are participating in one alpha helix are all next to each other in the polypeptide, right? They're local to one another, as opposed to distant ones on the other end of the polypeptide. They probably are also participating in secondary structure, but probably a different one. A different alpha helix, some different type of structure. Okay, cool. I lost my cursor. All right. Okay, so now turn my pen on before I forget, and we can get started with these questions. So our first question set is from the first part of the lecture, which is about the different levels of protein structure. As we just had a question about, some of them are more localized, some of them go across different areas of the protein, and we'll go into all of that as we go through and answer all of these questions. So first question is about the different levels of structure. So everybody take a few minutes, read over this question about what level of protein structure is described by each statement, and think about which level of structure applies to which one of these statements. All right, so can anybody tell me for the first one, number one, repeating local structures, which level of protein structure does this describe? Yeah. Secondary. Yeah, we just had a question about this before we got started. So this is B. For number two, which one describes the complete three-dimensional structure of a polypeptide? Yeah. Tertiary. Excellent. And for number three, the amino acid sequence, which level is that? Yep. Primary. And then our fourth one, the number and arrangement of subunits. Anybody got that one? The last one left. Anybody? D. Yeah, I heard it out there. Okay. So, just a quick word about nomenclature here. Was anybody confused between 2 and 4? So, in question 2, we're asking about a polypeptide, right? In question 4, we're asking about the number of subunits. So, a lot of times when I'm talking or when you're reading, you'll see used interchangeably the terms like polypeptide, you'll see sometimes a chain, you'll see subunit, so if we're talking about one polypeptide, that's the equivalent of one chain. and that is the equivalent of one subunit if a protein has multiple subunits. A single polypeptide is one long chain of amino acids that can have up to primary, secondary, and tertiary structure. To have quaternary structure, you're talking about multiple subunits or multiple separate individual polypeptide chains. In the case of a protein that is a monomer, that is, it only has one subunit, then the tertiary structure and the quaternary structure are the same. Because there are no further subunits to talk about for quaternary structure. For a polymer, you will have a separate individual quaternary structure. Yeah. Yeah, you could say that they're the same as each other. Yeah. Mm-hmm. Any questions about this? All right. So let's talk about the stabilizing interactions for each of these types of structure. So again, everybody take a couple minutes and think about how is each of our levels of protein structure going to be stabilized? What types of bonds? All right, so who can tell me how the primary structure is stabilized? What kind of bonds do we see there? Yeah. Yeah, covalent bonds. And so what is the name for those types of covalent bonds that we see? Yeah. Yeah, so we're seeing peptide bonds, which are a type of amide bond. So those are the bonds that are connecting together our individual amino acids in our polypeptide chain. That's our primary structure. All right, how about our secondary structure? Yeah. Hydrogen bonds and disulfide bonds. Does anybody agree or disagree with that? Yeah. Yeah, exactly. It's just hydrogen bonds. So when we think about our secondary structures, the types of bonds that help form secondary structures are only between the backbone of the amino acid. They're only going to involve hydrogen bonding in between the amino groups and the carboxyl groups of whatever amino acid they are sticking to. If you're talking about disulfide bonds, those are going to have to be between side chains, right? So, and we do not see bonding between side chains when we're talking about secondary structure. And we'll get more into that as we talk about the different types of secondary structure in the upcoming slides. And one of the reasons the secondary structures that we see are so universal among all proteins is because they are only involving interactions between the backbone. So every single amino acid has the same backbone, so every single amino acid is able to participate in these types of secondary structures that we're used to seeing. All right, how about our tertiary structures? What kind of bonds do we see? Yeah, we see A for sure. We see interactions among side chains in our tertiary structures. So we'll see definitely disulfide bonds, which are between cysteines. Any other types of interactions in our tertiary structure? Yep. E, yep, hydrophobic for sure. So when you have hydrophobic side chains, they'll interact using hydrophobic interactions. Yeah. Yes, it is all of them. That's absolutely right. So depending on what types of side chains are interacting, you'll see all of these different types of stabilizing interactions. So your disulfide bonds will be between cysteine residues. Your salt bridges can be between charged residues, like your acidic and your basic ones. Your covalent bonds, like we just heard, are a type of disulfide bonds are a type of covalent bond. So if you see disulfide, you're seeing covalent. Hydrogen bonds you'll see between some of our polar side chains. And your hydrophobic interactions you'll see among nonpolar side chains. What about our quaternary structure? What type of interactions do we see here? Yeah. Yeah, it's also all of them, because again, quaternary structures as well as tertiary structures are both interacting through their side chains. All right, any questions about these types of stabilizing interactions? Cysteine. Yeah, sorry. So cysteine is the amino acid that has that thiol group in the side chain. So that's the one amino acid that'll form disulfide groups between the two of them. Any other questions? All right, let's move on to talking about some of our secondary structure elements. So can anybody just shout out any secondary structure element they know? Alpha helix, yep, that's a good one. Any other ones? Yeah, exactly. We have beta, oops. Do we have any other types of secondary structure? Loops and turns, yep. So those are going to be found in alpha helices or beta sheets? Both in beta sheets, yeah. Any other types of secondary structure? I'm sorry? Yeah, there's some coiled, which we won't go too much into, but that is one. then the other option, which is sort of a non-option, is just unstructured. It's possible you might have some regions that are none of the above. So kind of the opposite of a secondary structure is just a non-structured or unstructured. And regions of the protein that are unstructured have a lot of flexibility often. So you'll see a lot of movement, different conformations. They don't stay still enough or have the right types of primary structure in order to form any one of these other types of secondary structure. Yeah. Are they inactivated? Not necessarily. Some proteins just have a lot of, not every single area of the protein is always participating in a specific structure. There are some areas of the protein that are like connecting to different regions, and those might be less unstructured. They might not always have a function. Those unstructured areas might not have a particular function they carry out. They might just be more structural, but it depends on the protein. Some proteins might change from unstructured to structured depending on a change from their environment, like a signaling cue or some other type of cue. So, sort of, I guess. Any other questions here? All right, so let's start by talking about alpha helices. So, first of all, what do we usually see? Like when you see a picture or a depiction of an alpha helix, what does it sort of look like? Yeah. Yeah, exactly. It's kind of like on a ribbon diagram, we'll see it drawn as just this sort of coil of ribbon. I won't try to draw it in 3D because we all know my drawing skills have not the best, but essentially you'll see something like this. I just drew a left-handed one because I'm left-handed. Here is a right-handed one, I think. I don't know. I guess you can't really tell based on my drawing. Anyway, so how many amino acids are in one turn of an alpha helix? So if I go from here up to here, that's one turn. How many amino acids have I progressed forward? Yeah, 3.6. Excellent. So yeah, having 3.6 amino acids there is what allows the side chains to kind of not get in each other's way. They can sort of be like staggered with each other as you're looking at the alpha helix, which sort of gets into the next part of our question, which is where do the side chains project in an alpha helix? Does it go towards the middle? Does it go radially away from, towards the outside? Does it go towards the end? Or are they on alternating sides? Yeah. Away, yeah. They go radially away from the center. So you can sort of imagine you have these side chains. I'm not drawing these accurately, but they're kind of pointing towards the outside. Okay, so how is that different from another? Are there other helices that we've talked about in class that that don't do this, that have their bases go towards the center? Can we think of an example of that? Yeah. DNA. Yeah, so this is the opposite of DNA, right? In DNA, you have a double helix where the bases go towards the middle and interact with each other in the middle of the helix. For proteins, we also have a helix, but in this helix, our bases, or in this case, our side chains, are on the outside. So that's just something to sort of think about as you're processing all this information. All right, so number 3, true or false? Amino acids in an alpha helix are contiguous in the polypeptide chain. Yeah. Contiguous meaning they're adjacent to each other. So amino acid 2, 3, 4, 5, 6, 7, 8, like all in order. So like in this alpha helix, this blue line I have drawn here, are all of the, if you're thinking about the primary structure of this polypeptide chain, are all the amino acids going in order down this helix, or are we skipping some? Are we going out of order somehow? Yeah, exactly. Like, yeah, it's not a trick question. They are. This is true. You have, they go in the order. So we don't skip any, we just go straight through. Every amino acid in the chain is going to be participating in the alpha helix. So can anybody describe the hydrogen bonding that is stabilizing the alpha helix? Where are the hydrogen bonds going? What are they connecting? Yeah. Yeah, exactly. So we're connecting together amino hydrogens to carboxyl oxygens, and they're every 4. So if this is the nth amino acid, this is the n plus 4th amino acid that it's connected to. And we can also think of that when we think about our weird little picture here, right? If we go 3.6 amino acids in every turn, your neighbor, so if this guy is number n, its n plus 4th amino acid is going to be about 1 level up this helix, right? And the reason it's a little bit off, if you have the n plus 4th, even though we have only 3.6 amino acids for every turn, that's because amino acids have, size, right? So you have your amino end, you have your carboxyl end, and you have your, this is not a great picture, but you get the idea, and you have your alpha carbon in the middle. So if you have the hydrogen on one, hydrogen bonding with the carbonyl of a different one, they're going to be offset by a little bit, right? Because this one will, they don't quite line up. So you don't get exactly 4 for every turn. They're lined up such that the aminos of one line up with the carbonyls of the other one. Yeah. No, yeah, you could also think of it as like n and n minus 4, or or it's just every 4th. It's not like it's not like a mathematical formula or anything. It's just it's the 4th one. Each amino acid is hydrogen bonding with the 4 amino acids later, and also the 4 amino acids earlier. It depends how you're counting it. Yeah, so like every one is sticking to every other one. It's so it's not like only every 4th are connected. Like they're all connected just to the one that's 4 away. If that makes more sense, hopefully. Kind of. Yeah, it's I wish I could draw it better. It's hard to imagine, but if you look at the picture from the slides, it makes it fairly clear. You can count better on that one. Yeah. Yep. The pitch. Yeah. Yeah, so the pitch refers to the height that's in between. So like if you're looking at this amino acid and that amino acid on this side, the pitch just refers to the height in angstroms. It's just a measure of size. Oh, that's a good question. I'm not sure. I can look that up. But yeah, I mean, it refers to something on the backbone, whether it's alpha carbon to alpha carbon or just amino to carboxyl. I'm not sure. But it's, yeah, just refers to the height. Any other questions? Yeah. 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 So yeah, your thumb is pointing. I really hope I'm not mixing this up, but I'm pretty sure your thumb is pointing towards the amino terminus. This is one of those things that like I look up before I say it every time because I always mix them up. But yeah, you can, if you imagine the curvature of the coil, I'm fairly sure, yeah, your thumb points towards the N terminus, and if your coiling is going this way, then it's right-handed. If your coiling is going this way, then you're left-handed. So like your coil is going in the direction that your fingers are going, and your thumb is pointing towards the N terminus. But I will double check that and look that up and correct it if I'm lying to you. Yeah. Any other questions? See some thinking. Yeah. Yeah, so if you think of your hand as like your thumb is the end and like the bottom of your hand is the C, the direction of the turning is your fingers. So if your fingers are turning this way, this is my right hand, but it looks like left to you. Yeah. Yeah, that's why I said I always mix this up. I will look it up and make sure. I believe your thumb goes towards the N terminal, but I might be wrong, and I will double check. What I said in the slides was correct. I just don't remember because, yeah, that's just how my brain works. Any other questions? Okay. So let's move on to beta sheets. So, yeah, so as we mentioned when we were talking about the types of secondary structure, we have parallel beta sheets and anti-parallel beta sheets. So, but either one, they're often, how do we usually draw beta sheets if you're looking at like a ribbon diagram of a protein? Yeah, I'm seeing a zigzag. Yeah, what else? Yeah, so sometimes we'll see a zigzag kind of like this. And that sort of shows you really well why it's called like a beta pleated sheet, because you'll have, side chains sticking out from either end, and it looks like it's folded. Another way you'll really often see them depicted is with those arrowheads, just like if you're looking at a ribbon diagram, so you'll see like an arrow, and like that'll tell you if some more information about the beta sheet that we'll go over as we go through this slide. All right, so parallel and anti-parallel beta sheets differ in what? Is it the N to C direction of the beta strands? Is it the arrangement of the side chains? Or is it the length of the beta strands? Yeah. Yes, it is the N to C direction. Yeah, so in this picture that I drew right above here, are those ones parallel or anti-parallel? They're parallel, yeah, so their arrows are pointing N to C, they're both going the same way, they are parallel. If I were to point them going the opposite way, these would be anti-parallel. And also, can anybody tell me the difference between a beta strand and a beta sheet? So in this question, we talked about parallel and anti-parallel beta sheets, and then we talked about the directionality of their strands. So what is, yeah? Yeah, exactly. So we have our whole thing here. So like in this picture, we have a whole beta sheet. And we have, in this picture, 3 strands that are making up this one sheet. Is that clear for everyone? Okay. Yeah, just because terminology can get confusing. So I wanted to make that very clear. So number 2, anti-parallel beta sheets are typically connected by blank, while parallel beta sheets are often connected by blanks or other structures. Yeah, want to do the first one? Yeah, so the first one is turns. So anti-parallel beta sheets can be straightforward to connect. They can, because of the directionality, right? If you start with this one on top and you're going to the next one on the bottom, you can do a very short little turn to connect together the two strands of that beta sheet. And because turns are often really short and they have these really harsh angles in them, we often see a couple of different amino acids in them. One is proline and one is glycine for different reasons, right? Proline already has a pretty sharp angle in it, so it likes to make little turns like this, so you'll find them in there. Glycine has basically the tiniest possible side chain. It's just a hydrogen, so it's easy to stick into a turn because there's not a lot sterically getting in the way there when you're trying to make that steep angle. And that's for turns. So what do we see often connecting parallel beta sheets? Yeah, loops. Yeah, so for a parallel beta sheet, if we have our strand on the top, if we want to connect to the strand on the bottom, we have to go all the way around to connect to the other end. So that's why we'll often see loops, which tend to be unstructured. They allow for some amount of flexibility. Or you can also see sometimes other structures that are in between the two strands of a parallel beta sheet. So number 3, true or false? Amino acids in a beta sheet are contiguous in the polypeptide chain. Our favorite question here. So we already have some pictures here of what amino acids are participating in our beta strand versus our beta sheet. So are all the amino acids in our beta sheet contiguous is my question. Seeing some shaking yes, some shaking no. All right, yeah, so the answer is no, it is false. And so you can see that pretty clearly in our illustration, right? So if you look at our parallel, we have our strands. So like our top strand, maybe that has amino acids, 2 to 8, and our next one has amino acids 25 to, 31 or whatever. And then the ones in between 8 and 25 are doing something else. They're part of a loop or they're hanging out somewhere else. They're not, you don't have a contiguous primary sequence of amino acids that are all participating to make up that beta sheet. Does that make sense for everyone? Yeah. Yeah, so like the amino acids that are physically forming the beta sheet are the ones that are in those strands that are drawn as those arrows. And then there are also amino acids that are part of, for example, that loop in the parallel one. Those are not participating in the beta sheet. They're connecting together the strands of the sheet, but the amino acids that are hanging out in that loop are not part of the beta sheet.
Okay, that's going to be like
So the numbering for a protein is always just given as the primary structure. So amino acid number 1 is the N-terminal one, and then you start counting from there, the way they're covalently bonded by those peptide bonds. So amino acids that have, so like in an alpha helix, it's straightforward. Every single amino acid in the primary sequence is part of that alpha helix. So you'll have, amino acids 10 to 65 in one alpha helix or whatever. But for a beta sheet, you'll have different strands are contiguous, and then in between them you'll have the breaks that are participating in loops or turns or other structures. Any questions about that? All right, so let's talk about number 4, the peptide backbone. So describe the hydrogen bonding that stabilizes a beta sheet structure that is part of the peptide backbone. So I gave away part of that, but we already talked about how only the peptide backbone is participating in stabilizing all of our secondary structures. So yeah, what can you tell me about that hydrogen bonding? Okay. Yeah, so let me draw another one because I kind of filled up my first one. So here's our parallel one. Here's our anti-parallel one. So yeah, tell me about the hydrogen bonds in one of them.
Parallel, they're actually straight down.
Mm-hmm.
They match up nicely.
Yeah. Mm-hmm. Yep. Yeah. So for just like in our alpha helix, our hydrogen bonding is going to be between the amino groups of one and the carboxyl groups of another. And so, like you said, for the anti-parallel ones, those hydrogen bonds are lined up such that they're all parallel to each other and they form these nice straight lines up and down. That makes a very stable, happy structure. For the parallel beta sheets, the hydrogen bonding angles don't quite line up the same. So they are not quite linear. They'll look a little bit kind of off kilter, if you will. So they're still pretty good. They're close to linear, but they're not exactly linear. So this means that parallel beta sheets are slightly less maybe stable than an anti-parallel one. They're both extremely stable, but parallel ones have slightly less ideal hydrogen bond angles. So can anybody tell me where are the side chains in a beta sheet? Yeah, I'm seeing some hand gestures to the side. Yeah, exactly. So on a side chain or on a beta sheet, the side chains are off of either face. So it's sort of easier to see on this sort of zigzag drawing up top, but you'll see side chains alternating. So every other one will stick out the front or the back of this sheet, right? And that's where you got that sort of pleated appearance with side chains sticking out of each kind of fold in your pleat. All right. Any questions about, yeah. Okay.
But when we look at pictures of the parallel beta sheets and their hydrogen bonds, we see that the bond between the nitrogen and the hydrogen is fixed, as we kind of did in the slides. But we also kind of knew that when you have a hydrogen bond, you kind of have it in a straight line, and we don't have that.
Yeah. Yeah.
How does that work?
Yeah, so I mean they're very close to linear. Like the way I'm drawing it is kind of exaggerated. Like they're not so far off that they do essentially have hydrogen bond character because they're very strong and they're very, very close to linear. They're just not perfectly linear. So they're a little bit, you could say the interaction is a little bit weaker than a really nice linear hydrogen bond. It's close, but
It's close enough that it works.
yeah, exactly. Any other questions about beta sheets? All right. So let's talk about tertiary and quaternary structures. Yep. Okay. Usually oxygens are drawn in red. I'm having trouble recalling the exact picture you're talking about. Let me see if I can pull it up. Okay. No, this is my wrong PowerPoint. This guy.
Like on the right, the black ball on the right.
Here.
The right.
Mm-hmm. Yep.
And then what's on the right?
This is the alpha carbon.
Did I show that one?
This is, it's not, yeah, so this isn't terminal. This is like a snapshot of like the middle. Yeah. So you're assuming that it extends further in either direction. I mean, it's just not showing the C terminus. The polypeptide keeps going. This is just sort of like a snapshot of the middle. So like, for example, I mean, this is meant to depict one single polypeptide that's forming a beta sheet, right? But it's not showing the loops and turns on either end that connect it to the rest of the protein. So it's just sort of like it pulled it out and grabbed it. So we're not actually seeing any of the termini here. We're just sort of, we grabbed an arbitrary section in the middle that's part of a sheet. That's part of a beta sheet. I mean, you can. Yeah, well, I guess, I mean, here you're not, you can tell which direction it's going based on the actual structure of it. So you know it's going, in this case, N to C from right to left. But you wouldn't say either end is the N terminus or the C terminus in this picture. They're not shown. That's the directionality of the amino acids. So if you, this is the amino alpha carbonyl, amino alpha carbonyl, amino alpha carbonyl. So it's the way amino acids are drawn conventionally is, or like the other way, right? So this N to C one, you have amino alpha carbonyl going that way. So from amino to alpha to carbonyl, that's N towards C, right? So whichever way your N to C is going, it's either going to the left or to the right. And you can assume that somewhere all the way on the right at the top, your actual amino terminus is there, but it's not shown. Exactly, yeah. So you know which direction it's going based on the backbone that you can see. Yeah, exactly. So like you can see this blue one, it's the amino, and so it's hydrogen bonding with the oxygen from the carbonyl group from the one below. Yeah. Yeah, and the directionality is what makes these hydrogen bonds really nice and linear compared to these hydrogen bonds in our parallel, because now we have N to C going the same direction. The geometry is just going to line up a little bit differently because of that. All right. Any other questions about that? All right. So let's talk about tertiary and quaternary structures. So everybody take a few minutes and answer these true or false questions, and then the last question about our multimeric protein. Okay, so let's start with the first one. So tertiary structure refers to the structure of a protein with 3 subunits. Yeah, false. So can we correct that statement? Tertiary structure refers to... Yeah, so it refers to the 3D structure of a subunit. Alright, the second one, quaternary structure, refers to the structure of a protein with 4 subunits. Yeah, also false. So what does quaternary structure refers to? Okay, yeah, go ahead. Yeah, so it refers to the arrangement of multiple subunits. It doesn't have to be multiple, yeah. It doesn't have to be 4, that is true. It often refers to the structure of the arrangement. And I'll say often. Not all proteins have multiple subunits. When they do have multiple subunits, that is what the quaternary structure refers to. Okay, so third one. Domains are a part of the tertiary structure of a protein. Seeing some nodding, some blank stares. Alright, yes, that is true. Can anybody tell me what is a domain? Yeah. Yeah, exactly. So domains are often folded independently. They often have their own function. But they're part of the tertiary structure, right? So they're part of that one polypeptide chain, might have multiple domains on it that are, when they're translated, they're all translated together as one single chain, but they might fold separately. They might interact with different parts of the cell. They might carry out different functions. All right, so do you have a question, or do you want to answer the last one? Okay, go ahead. Yeah. Yeah, it kind of depends on the protein. So for a monomeric protein, it kind of stops there at tertiary. So you could consider those types of groups as part of the tertiary. For a protein with multiple subunits that has quaternary structure, you could also consider it as part of the quaternary structure. It's a little bit ambiguous. You could just sort of say like they apply to the highest level of structure that protein has. If that's an easier way to think about it. Yeah. Okay, so for the fourth one, the quaternary structure requires covalent interactions between polypeptide chains. Yeah. Excellent. Yeah, it doesn't require. You can have covalent interactions, but they're not required. So when would you see covalent interactions as part of quaternary structure? Anybody? Yeah. Exactly, yeah. So you could have but that's not the only way to connect together subunits, right? We can also have all those other non-covalent interactions that we talked about a few other slides ago. All right, so let's go to the second part of this slide. So a protein that contains 3 different polypeptide chains is called a a heterotrimer. So we have hetero, that means they're different, and we have trimer, which means 3 subunits, right? So it is a hetero trimer. You could use homo instead of hetero if all of the subunits are the same. So if you had 3 subunits, all 3 were the same, it would be a homotrimer. If you had 3 subunits, all of them are different, it's a heterotrimer. If you have 3 subunits, 2 are the same, 1 are different, still a heterotrimer. All 3 needs to be the same if you want to use the homo prefix. And then for the ones in the bottom, it's just how many subunits you have. So monomer, 1 subunit, dimer 2, trimer 3, triad is not a word we use here, and tetramer is 4. More than that, more than 4, we'll often just stop counting and we'll say oligomer or polymer to mean a bunch. Any questions about naming, this kind of general kind of naming? All right, so let's start looking at some structures of real proteins. Oh, do you have a question?
Yeah.
About this?
Yeah, about that.
Okay.
The great endpoint stuff that's going on there. Also, the chains.
Yes, these are all parts of the protein. Yeah. There are different depictions of the protein, but yeah, this is all protein here. So everybody take a few minutes and squint at this picture and see what you can see. I wonder if I can blow this up a little bit. Kind of. It's fun. I can blow it up, but then you can't see the whole thing. Would you rather I keep this like area blown up, or do you want me to zoom out? Doesn't matter. Okay. All right, everybody take a few minutes then. Alright, so does anybody want to tell me about any kind of structure they could see and identify somewhere in this diagram, in this picture of our spike protein? Yeah. It is a trimer. It's actually a homotrimer, but it's hard to tell from this picture. Yeah, so what level of structure would that? That's quaternary, yeah. So what are the three components of our trimer?
The one that we see in every catalog.
Yeah, exactly, yeah. So we have this kind of rainbow detailed one. And then they're sort of showing that the other two trimers are the same. They just have them in white and gray. That's 3 subunits. We got a trimer. All of the subunits are the same as one another. It's a homotrimer. Yeah. They're part of one single polypeptide chain. So the different colors in that rainbow one represent another type of structure that I won't give away. But the I mean, it's really hard to tell just from the picture that they're all connected, but they are. Just from this picture, you can sort of see, okay, I see a gray blob, a white blob, and this fancy rainbow thing. So, like, for me, just looking at it, not knowing anything about the spike protein, I see, okay, the gray and the white, definitely 2 different subunits, and, like, your educated guess is the rainbow one kind of looks similar to the other 2. It's probably the 3rd subunit, but pointing out all the details of the structure, of a different level of structure that we can get into. So does anybody have any other types of structures they want to point out? Yeah. Yeah, exactly. Yeah, so right over here, let me see if I can zoom in so everybody can see what you're talking about. But we have these nice little anti-parallel beta sheets in green. And you can see that because the arrows are pointing in opposite directions from one another. And so what level of structure is that? Secondary. Over here. It's not structured enough that I would call it a beta barrel. It's kind of like it wishes it was a beta barrel but didn't get there yet. But yeah, you're right. It has some beta sheet character to it, and I can't even tell if they're parallel or anti-parallel, honestly. But yeah, that there is some beta sheet character there, and then there's a lot of unstructured character, right? So we have certainly some unstructured reasons. It's kind of like a gray area. Like over here, like these are pretty short. I would say that's a loop, although the loop itself is not structured, right? Like it happens to be drawn in this particular exact shape, but you know, it might bend over a little bit. It might start curving or bending around in whatever ways are favorable, especially if it's in solution. And that's what makes them unstructured. They have a primary structure, but they're not in a rigid secondary structure. Yeah. 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 You could say either. Over here, it's sort of unstructured. You can say it's a loop if it's connecting 2 different beta strands, which in this case they are. So like you could say both. Yeah, sorry, that's not a great answer. Yeah, it's sort of one of those things where it kind of depends, or someone might say one thing, someone might say a different thing. Any other levels of structure that we're seeing here? Yeah. Yeah, so in our tertiary structure, we have some domains. So the domains, they try to point out here by making the domains different colors. That's what we're seeing on our rainbow diagram. So this green area is one pretty clear domain. This blue area is another pretty clear domain. The red and the yellow are kind of all mushed together. It's hard to say they're not really separate from each other in space, but they are trying to mark them out as maybe different domains. Maybe they carry out different things or move differently. This purple area on the bottom looks like a domain to me. Yeah. What else do we see? Yep. So yeah, so for like the yellow and the red one down here, I agree. I wouldn't necessarily say that's like a whole domain. That's really just like a little piece. And there's different color ones kind of grouped on top of each other. So maybe that whole thing, like the red and the yellow and the orange, maybe are sort of one domain. It's very clear. Like I would say for sure, like these are very clear as domains. What? So unstructured is part of secondary structure. Domains are part of tertiary structure. So domains are groups of secondary structure, and domains often have a separate function that they carry out. So this green region of the protein, it's a domain. It has some beta sheet character and some unstructured character as a secondary, and it has some kind of function that it carries out as a domain, like in the cell. Does anybody see any other things we haven't mentioned here? I know somebody said, but we didn't actually write down, there's also alpha helices here, right? What level of structure do those go under? Secondary, yeah. So we can see this nice yellow one here. There's a nice red one right next to it. And those are really in that one particular region of the protein towards the bottom in this drawing. And it might help also to start looking at our next slide, right? So our next slide has the same picture that we were looking at on the previous slide. It also has it from a different angle. So this is kind of looking at it from the top down. And from the top down, you can see more clearly how those trimers are sort of connected to each other, how the gray one is hugging the white one, which is hugging the kind of rainbow one. And there's also this drawing at the top. Does anybody want to say sort of like what this type of diagram is showing us? Yeah. Yeah, exactly. So this is sort of a view of the protein that starts with the primary structure. So we have the order of amino acids. Number 1 is over here. Number 1273 is over here. And then as part of the different, or at different locations along the primary structure, we see in colors the different domains of the protein. So these are different regions of the protein that are going to fold independently and maybe have independent functions. And these domains are often named. So one domain on the spike protein that you may have heard of is this one that's shown in green here, the receptor binding domain, or the RBD. This is the part of the spike that's actually going to interact with the ACE2 receptor on your cell and get inside your cell. And that's one particular domain of this protein that has one job. In blue, all the way on the left, we have the N-terminal domain because it's on the N-terminal side of the protein. And that one is shown in blue, kind of on the side over here, and that has its own function and does its own thing. Anybody have any questions or thoughts about this? All right. One other thing I forgot to point out or ask. So we talked about, as part of our tertiary structure, we talked about, yeah, go ahead. Yep. Mm-hmm. Yeah, so that's what we were talking about before, how kind of the red and the yellow and the orange are all kind of actually physically overlapping in 3D also. And the way it's drawn on this diagram that shows the primary structure, you can see they're also just sort of all overlapping with each other. So that is just how this particular protein is structured. Not all proteins are like that. A lot of times you'll see very distinct domains that are very obvious. This particular one has sort of 3 or so different ones that are all kind of mushed together for whatever reason. Their functions are probably related to one another. I don't know off the top of my head what each of those ones does. So one last thing I forgot to mention or review as part of the different levels of structure is another aspect we should look for when we're looking at tertiary structure, which is motifs. So does anybody see any motifs in here? I know they're hard to see because this is kind of small, but if you squint down, are there any really nice looking motifs that anyone can see? Yeah. Yeah, so like it's a bunch of alpha helices, which are parts of secondary structure, but they're like specific arrangements of secondary structure that fall under motifs. Yeah, go ahead. Yeah, a beta hairpin. Yeah, where do you see one of those? I'm sorry? In green. Yeah, since we do have an anti-parallel sheet up here in green, you might see, it's really hard to tell from this picture. I actually don't think there are any beta hairpins on the green one. There is a really good beta hairpin that I saw actually on the bottom in purple. If you look right here, which is, yeah, no, I can't draw because I'm zoomed in, of course. But if you see, let me circle it first and then we'll zoom back in again. Yeah, so if you see I have circled in blue there, there's a, there are 2 beta strands and they're connected by this very short turn that helps them change direction very quickly in only a few amino acids. So that's an example of a beta hairpin, which is sort of another name for a turn. In case, and it's really hard to see in this picture, all the individual types of motifs, but as a review, we can look at our motifs diagram. So we have, these are our 4 really common motifs that we see as part of tertiary structure. So here's our beta hairpin that we just saw an example of in purple. We also have beta barrels, which someone else asked about, which we don't see in the spike protein. We don't really see either of these in the spike protein, that's very clear. There may or may not be an alpha beta or a beta alpha beta, but I didn't see one. There might be one in there that I didn't see, but definitely you see some good examples of beta hairpins in the spike protein though. Yeah. You could. Mm-hmm. As long as they're interacting with each other via side chain interactions, yes. I would say you'd you'd be less likely to see that across different domains because domains are often folding independently, so they're mostly interacting with other amino acids that are in that same domain. But I'm sure that's not always true. I'm sure that there are times when in between different domains you'll see these types of tertiary motifs. Any other questions about our levels of structure? Okay, so let's talk a little bit more about amino acid side chains and where we're going to find them in a protein. So everybody take a minute or two and think about these 5 amino acids and tell me if they're more likely to be solvent exposed or if they're more likely to be buried somewhere inside the protein. All right, so what do you have to know about an amino acid to tell whether it's going to be exposed or buried? Yeah. Yeah, exactly. So what categories of amino acids are going to be solvent exposed? Yeah. Yeah, so more hydrophilic. So what types of side chains would that be? Yeah, polar or charged. And what about our buried amino acids? What are they going to be? Yeah, hydrophobic ones, which are our nonpolar. All right, so let's go through each of our amino acids here. So arginine, exposed or buried? Exposed, yeah. Yeah, is arginine polar? Is it charged? Yeah, so yeah, arginine is often charged. What about asparagine? Exposed, yep. And asparagine is polar. What about isoleucine? Yeah, it is buried. Yeah, it's fairly large and it's nonpolar. What about glutamic acid? Yes, very polar. It's usually charged. It's therefore solvent exposed. And what about phenylalanine? Yeah, varied. Yep. Yeah, this is our bulky nonpolar. Yeah, so for these ones, it's all fairly obvious, right? They're like charged, which makes them very happy to be in water, or like very large and nonpolar, which makes them very unhappy to be surrounded by water. So, or makes water unhappy to be around them. Either way, they don't like each other. For some other amino acid side chains, it's maybe less obvious. So, one example is something like alanine. Alanine is nonpolar, right? But it's really small. So, if you have an alanine residue, it's probably happy either way. It's happy to be buried inside because it has that hydrophobic character. But it's also not too unhappy to be solvent exposed because it's not super duper nonpolar. It's kind of, I wouldn't say it's neutral, but it's, of the nonpolar ones, it's not as unhappy about it. All right, so let's take our last little bit of time and talk about our Ramachandran plot. So, yeah, question number 1, what do these axis labels refer to? Yeah. Yeah, these are dihedral angles. So are dihedral angles the same as bond angles, or are they different? Yeah. Sort of. They are different from each other. They do refer to one angle. So let's draw a picture. I think that's the easiest way to think about it. So let's say you have 3 different atoms, right? A, B, and C. They're connected by covalent bonds. So if you have the angle between these 2 bonds that are adjacent to one another, that's a bond angle. Right? It's 2 bonds that are right next to each other. It's connecting. We have one central atom in between. So something like water. We've talked about our bond angles in water in this class before. Dihedral angles are a little bit different because for a dihedral angle, we're talking about the angle between bonds that have another bond in between them. So for a dihedral angle, we need to talk about 4 different atoms that have bonds between them. And now we're talking about the angle that has to do with the rotation about these bonds. So like, this is much harder for me to draw on this slide, but like this rotation like in space about this bond essentially. That tells you the angle between this and between this. It is, yeah, not my greatest picture, but I do have our little summary slide from the lecture that has a better picture of that. So in the case of a dihedral angle, we're talking about these non-sequential bonds. So we have this is the example of our phi angle, right? So we have our amino grouped alpha carbon in the middle, and then we have the angle between these bonds drawn in yellow and in blue that are not sequential, right? There's a bond in between them, and we're talking about the angle between these 2 bonds that are outlined in blue and yellow. It's a little bit hard to visualize. If anybody still has their little ball and stick model kits from organic chemistry, I highly recommend setting this up and helping yourself visualize it better because it's confusing to just picture when you're seeing it flat on a page. that being said, yeah, so these axes refer to our dihedral angles. And there's two different dihedral angles because we have, for every amino acid, we have two different available locations where we have rotation about a bond, right? So we have phi and psi. So even though, and then of course we have our one bond that does not allow rotation, right? So which of our bond is giving our amino acid this rigidity that doesn't allow rotation? Yeah, the amide bond, yeah. And why is that? Why can't we have rotation about our amide bond? Exactly, yeah. So due to resonance, our amide bonds have partial double bond character. They do not allow free rotation like our other single bonds. So that's why we have just those two dihedral angles in each of our amino acids. All right, so getting back to our plot, what do the blue and the green areas on this plot in the sort of background, what do those tell us? Yeah. Yeah, exactly. So blue is favorable or optimal angles. Green is acceptable. So if you see a protein and you plot out all of the different phi and psi angles in a protein, you will see all of them falling mostly in the blue region and also in the green region, and very few, if any, in the sort of beige areas. So let's talk now about these boxes. So what is our red box? What type of structure is that? The red box? No, it is not. Sorry. Yeah. Yeah, so the red box is our anti... What about our yellow box? The parallel beta sheets. Yep. Wow, that is impossible to see. And one way you can sort of help yourself remember those is that these two boxes are in the region of this plot that is near 180 by 180, or negative 180, because negative and positive 180 are the same, right? So if you think about 180 degree dihedral angle, that's pretty close to planar. And we know that our beta sheets are pretty close to planar, flat planes. So anytime you have any kind of beta sheet character, your dihedral angles are going to be closer to 180 for both of them. So that brings us to our third box, our purple box. So what does that represent? Yeah, that's our alpha helix. And so you can remember that as the one that's not planar. You don't have to worry too much about the details of like the exact numbers, but sort of know approximately where your these different secondary structures are. And I should mention for the purple box, our alpha helix, is that our right-handed ones or our left-handed ones? It's our right-handed ones. So does anybody remember where our left-handed ones were? Yeah, I see pointing, which is unhelpful. But yeah, so it's over here, right? This is where our left-handed. So those are also acceptable. Maybe not as favorable as our right-handed ones, but you do see them occasionally. And does anybody know what this region represents all the way on the bottom? Yeah, exactly. So this piece on the bottom is actually just kind of the wraparound from the top, right? Our 180 is the same as our 180. So this is just the overlapped, or the remainder of our beta sheet kind of region. So does that make sense for everyone? So like if you could sort of imagine this piece on the bottom, it's just kind of like an extension of this little island at the top. It just went past 180 and reached around back to the negative 180, if that kind of helps. All right. So let's leave it there for today. We'll pick up a little bit more with Ramachandran plots on Thursday, and hopefully we'll get through the rest of chapter 6. I'm going to try to have chapter 7 up either like tonight or tomorrow, so you can get started on chapter 7 if you'd like. And for your exams, I am not completely done grading them yet, and we're still waiting on a couple people who are sick to make them up. So I won't be returning them to you this week, but I will hopefully get your scores to you this week. But I do not have them ready yet for today. And also, if anybody wants to see me after this, you can either chat with me right after class or go to my office hours. And I posted it on Brightspace, but I do have a different office now. I'm in the 3rd floor of the Natural Sciences in room 3040, and that's posted on Brightspace. Yeah.
I still can't picture this at all.
Okay.
I don't understand.
Hold on one second.